Good afternoon, everybody. I'm Mary Cooloff, the President of the Royal Society of Tasmania, and it's my pleasure to welcome you to today's webinar. In beginning this webinar, I pay particular respect to the traditional owners of the land upon which we meet today, the Muwinina people, and pay respect also to Tasmania's Aboriginal community who have ongoing custodianship of this land. The Society pays respect to elders past, present and emerging. We acknowledge that Tasmanian Aboriginal people have survived severe and unjust impacts resulting from invasion and dispossession of their country. As an institution dedicated to the advancement of knowledge, the Royal Society of Tasmania recognises Aboriginal cultural knowledge and practices and seeks to respect and honour those traditions and the deep understanding they represent. Today's speaker is Dr. Jessica Ericson, who is joining us today from New Zealand. It's a great advantage of presenting our lecture series this year by webinar, in that we would otherwise probably not have been able to hear Jessica speak. And Jessica was one of our Doctoral Award winners for the Royal Society Doctoral Award 2019. Last month we heard our other winner speak, Dr. Indrani Mukherjee, who gave a terrific presentation, and I've had a sneak peek of Jessica's talk today, and I can tell you that you're in for a treat today as well. So a couple of things about today's webinar. You're probably wondering if you'll be able to ask a question of Jessica at the end of her talk, and yes, you will, by using the Q&A button at the bottom of your screen. Just type your question in, and I'll be able to see that, and I'll be able to ask Jessica a selection of those questions. Today's lecture is being recorded, and it will be available later on our Royal Society of Tasmania YouTube channel. If you've missed any of our previous lectures this year and you're interested, please go to that channel and you can watch them all. And our website will be on the screen a little bit later in the presentation so that you can find the link to the YouTube channel from that. Our Royal Society Doctoral Awards are selected annually for excellence in research in any field, for research that has been largely undertaken in Tasmania or under the aegis of a Tasmanian-based organisation. And I'd like to mention that our Doctoral Awards for 2020 are still open. So if you know an outstanding young researcher, you still have a chance to nominate them. Nominations close on the 15th of November. We're very pleased that so many members and supporters are able to join us today. And now it's my pleasure to tell you a little bit about today's speaker. Dr. Jessica Ericson specialises in the study of ecologically and economically important marine invertebrates from temperate and polar regions. Her research focuses primarily on how climate change stressors, for example, ocean acidification and warming, affect the physiology, biochemistry, immunology and behaviour of different invertebrate species. She has studied a wide range of charismatic microfauna, including limpets, mussels, oysters, sea urchin and krill. The unique responses of these organisms to their environment always provide opportunities to learn something new. Her studies have taken her to remote locations, including Antarctica, and she has spent many weeks at sea on research voyages. Jessica was born in New Zealand and comes from a long line of lighthouse keepers and scientists. So she believes it is no coincidence that she chose marine science as a career path. Jessica is also a keen mountain biker, ocean swimmer and printmaker. She lives in Nelson, New Zealand, but considers Tasmania to be her second home. And today, Jessica is going to talk to us on the topic of Antarctic krill. What do the Southern Ocean's charismatic omega-fauna eat, and how might they fare in a future high CO2 world? Thank you, Jessica. I hand the screen over to you.
Thank you very much, Mary, for the introduction. And thank you to everyone who has tuned in on your precious Sunday afternoon. Firstly, I'd just really like to give a sincere thank you to the Royal Society for giving me this award. I was really elated and surprised when I found out that I'd received the award, and it was actually a couple of days before my PhD graduation in Hobart that I found out. So it was a real icing on the cake for me to travel to Hobart knowing that I'd also won this award. And secondly, I just want to thank you so much for the opportunity to talk today. As Mary said, it's a shame that I can't be in Tasmania, but this is just the world that we live in at the moment, obviously. But it's great, actually. I don't know about everybody else, but I've really been enjoying these webinars at the moment, and the fact that people can tune in from anywhere in the world. So as you can hear, and Mary said, I'm a New Zealander. I'm currently living in Nelson in the top of the South Island, and I work for a company called the Cawthron Institute, and that's a New Zealand independent science organisation specialising in marine and freshwater research. So I made the move over to Tasmania in 2015 to do my PhD, and I spent 3.5 wonderful years in Tassie with my husband Nick. And hopefully I'll be back as soon as I can to visit, because we absolutely loved it there. Today I'm going to take you on a journey through some of my PhD and into the wonderful world of this animal here, the Antarctic krill. Throughout my PhD, I was lucky enough to work with a range of institutions in Hobart that are really world class in terms of the research. So some of them are listed here. We've got the University of Tasmania and the Institute for Marine and Antarctic Studies. Many of you have probably seen that building on the waterfront in Hobart. And also CSIRO, which is next door to IMAS. So it was really nice. I could walk backwards and forwards to do some of my lab work at CSIRO. And also the Australian Antarctic Division in Hobart as well, where I did some of my lab experiments on krill that you'll hear about soon. So just to begin, I'm going to give you a bit of a brief background on who I am and what I do. So sometimes I feel like a bit of a walking nature versus nurture science experiment, really, or with nature or genetics playing a huge role in basically the career path that I've chosen. So here's those lighthouse keepers that Mary mentioned. On my father's side, both my nana and my papa, both of their parents worked in lighthouses all around New Zealand. And so this lighthouse is one of those ones, and it's actually not far from where I live right now, which is amazing. Then I've got my great-grandfather, John Power. He was a fellow of the Royal Society of South Africa, and he was appointed a fellow of the Royal Society in 1931. So I actually only found that out after I received the Royal Society Award. So that was pretty cool to find out. And he was a reptile scientist. And my dear granddad, Ron Power, he was a very talented entomologist, and he actually worked in Nelson too, where I live at the moment. So pretty interesting series of events, and you can see why I've become a marine scientist today from basically my background or my genetics. And so my research has 3 main themes. I specialise in studying the physiology and biochemistry of marine invertebrates from temperate and polar regions. So I have been lucky enough to visit the Antarctic during my research. It was amazing. I didn't actually go there for my PhD. As you'll see, I was very fortunate to receive all my samples for that research in Hobart. But I've been to lots of exciting places. I'm very lucky. This is a photo of me looking like some kind of action hero dressed up to go snorkeling in Antarctica to collect some animals. And on the bottom left here, some of the different species that I've worked on. So you'd be fooled into thinking from all these pictures that I spend a lot of time in the field, but I do spend most of my time in the lab. This picture of me here at my current job. But I like to do both. I love to go out into the field and work in the lab, because when you go out into the field, you really get a perspective of how your research applies to the real world. And then in the lab, you get to really nail down some of those things you specifically want to look at. So it's really exciting. I also work alongside fishermen, marine farmers, and other scientists. And another theme I've kind of realized lately that my research follows is that I study a lot of organisms that are nutritious for their higher predators, including humans. So that's how I came up with this term, charismatic omega-fauna, because you've probably all heard the term charismatic megafauna before. So for example, whales and penguins and seals. that everyone is very familiar with. And I work on smaller things, but they're not really microfauna either. They're not really, sometimes they're microscopic, but they're often just kind of smaller animals. So I've come up with this term, omega-fauna, because a lot of the species that I study are very nutritious and full of omega-3 fatty acids, which are very nutritious for their predators. So I was trying to decide how I can describe a PhD to people, and it's really difficult to describe 3.5 years, a 3.5 year experience. But I thought it was kind of uncanny how much a PhD can sound like an Antarctic book cover. So, for example, the worst journey in the world, the impossible first, endurance, the coldest March, or maybe the coldest March, April, May, June, July, and August if you're doing a PhD in Tasmania. And a PhD can be all of these things at some time during the journey. But I was looking at these book titles and thinking they didn't really describe my PhD experience that well. And the book that I thought described my experience is this book called An Unsung Hero. So my PhD I really loved. I had a great experience. And a lot of that was to do with a lot of the unsung heroes behind the scenes that helped me out along the way. And when you finish your thesis, you kind of have all the glory, but really there was a big team helping me behind the scenes that really made it successful. And here they are. So I actually had 5 supervisors, and anyone who's listening who's done a PhD before might think, oh, that sounds like way too many people. They'll all be putting their 5 cents worth in and it'll be hard to get anywhere. But no, I just had a great team. It was we just worked really well together. And I was really fortunate to have that perspective from all those different people, and they're all very humble people, and I know some of them are watching right now. But lots of people would count themselves lucky to work with just one of these amazing scientists. So I was fortunate enough to have all of them and have that knowledge passed down to me from all those people. And I had Nicole, who's pictured here in the lab. She was also working on her PhD along at the same time as me, so her research was funded by the same grant. So it was really nice to have that person who really knew what you're going through or what had to be done. And of course, my family and friends who kept me sane and happy and sat through practice presentations, including this one. My husband, Nick, had to sit through this one already. They all celebrated my wins with me, and the mountain bikers of Hobart were really great as well, because they were just there to keep me doing my hobbies and having fun. So I guess if I'm going to pass on any pearls of wisdom to any other PhD students, my advice would be that you don't have to be the brightest crayon in the box to have a successful PhD experience. You mainly just need perseverance, optimism, support, and balance. And I think balance was the key thing for me, because my supervisors helped me to have that balance between high risk and lower risk research that I think everyone should have in their PhDs. And then I had that balance of family and friends to keep me happy and really made everything a success. All right, so away from the human unsung heroes and onto the real unsung hero of the story, and that is the Antarctic krill. And I think the unsung hero does describe them quite well, actually, because they're pretty amazing and they really don't get enough credit. So despite what you might think, Antarctic krill are actually not that small, and they're not that tiny. So they can weigh up to 2 grams, and they can be up to 60 millimeters in length. Their diet consists of mainly microalgae, and you can see that here in their digestive gland. And you can see their feeding legs here. So they kind of act in a similar way to the baleen and a whale. So they kind of filter through their food, and they can sort out particles and consume them. They have a really muscular tail, so scientists used to think they would drift along in the currents and kind of passively, but in actual fact, they can they have these really strong tail and strong swimming legs that they can use to swim against the current and propel their bodies, so they don't just drift around passively. And a key feature of euphausids, so krill, is that the gills are actually found on the outside of their body. And that's because they have a really high requirement for oxygen consumption, because they swim really quickly and they're found in quite packed, dense swarms. And having these gills on the outside of their body allows them to extract oxygen most efficiently from the seawater. All right, so this is the part of the talk where I'm hoping to convince you that krill should become your favorite animal. So they've actually got one of the most abundant, they're one of the most abundant animals on Earth in terms of their biomass. So their biomass relates to the number of individuals multiplied by their average weight. So you can see that cattle come in first place at 520 million metric tons. Krill actually come in second at 400 million metric tons. And humans are actually third with 350 million metric tons. Krill can grow and shrink, and that's dependent on their food supply and their metabolic rate. So when they come into the winter months, they will often shrink in size and depress their metabolic rate, and we'll talk a bit more about that later on. But that makes it hard to determine their age. So scientists used to think that if you measured their length, they would think that larger krill are older krill, but that's not necessarily the case. They live in seawater temperatures between minus 1.8 degrees and 5 degrees in the Southern Ocean. They have a really wide vertical and horizontal distribution, and they've been found at 4.5 kilometre depths. So now that we've got some pretty amazing technology and we can send remotely operated vehicles down into those depths, they've actually been able to see krill down there, which is pretty amazing. And this is the fact that blows my mind. They live in huge swarms, up to 3 million metric tons of animals that can stretch for 20 kilometres. So it's really hard to visualise that. And some of these swarms can be kind of 200 metres in depth as well. Some aspects of krill physiology are partially controlled by a biological clock system. So this is a system that is essentially turned on by a cue. And so for Antarctic krill, that we think that might be the light cycle. So in the Antarctic, there's basically 24 hours daylight in summer and 24 hours darkness in winter. And then in autumn and spring, you have kind of intermediates of that. But it's really interesting that some of their physiological processes, they will start to turn on or off depending on the light cycle. And this last fact is kind of the one that underpins a lot of my PhD research, and that's that krill are a highly nutritious food source for their predators. So on the left here, you've got a Southern Ocean food web. Obviously, images are not to scale. Krill are quite large, but they're not that large. And you can see that krill eat zooplankton, bacteria, viruses, protozoa, and phytoplankton. And importantly, almost everything else in the Southern Ocean eats krill. So you've got whales, penguins, you name it, they all feed on krill. And it's this lipid energy in krill that fuels the Southern Ocean ecosystem. So lipids are basically the building blocks of fats and oils. So when I'm talking about lipids, you can think of basically fat. And the krill themselves are highly nutritious due to their main food source, which is phytoplankton or microalgae. And these phytoplankton are rich in long-chain omega-3 polyunsaturated fatty acids. So that's a bit of a mouthful, but many of you have probably heard of these before because they're in, they obviously make very healthy for humans, and they're in those nutraceutical products, fish oils and things that you will be familiar with. And you can think of fatty acids as the biochemical building blocks of lipids. So many of the fatty acids are passed up through the food chain with minimal modification. And so they can be used as dietary biomarkers, and they complement other approaches such as gut content analysis. So these are some of the things that krill feed on, and the underneath here are the fatty acids that these organisms are rich in. So if krill have been feeding on a lot of diatoms, for example, you might see higher levels of these 2 fatty acids below. And so we knew a bit about krill diet already, but the data was patchy. So most of the data was collected in summer. And you might have we had some different seasonal data for the krill diet, but it was across different years. So you might have a smaller amount of data for, say, summer in, let's say, 2004, and then you might have some winter data in 2010. So it was kind of quite patchy. And so my aim in my PhD was to investigate the diet of krill using an unprecedented high resolution of samples. And we were able to do this with the help of the krill fishery. So why use samples from the fishery? Scientific research in the Antarctic is, as you can imagine, very expensive. And it often only occurs in summer because it's obviously very hard to get a ship down to the Antarctic when there's a lot of sea ice around. So in winter it becomes very difficult. And you're also competing with a lot of other research when you're down on an Antarctic research vessel. So you've got a lot of oceanographers and other people doing other things as well. So this is a picture of Molly. I never met Molly, but she did her PhD before me. And this is her collecting krill for her PhD using this tiny little net. You might not even be able to see that very well. So really exciting way to gather samples for your research, but obviously you can't get too many samples that way. So if you cross over to the right here with the krill fishery, they operate in Antarctica almost year round. So apart from a couple of months where in spring. And because they're fishing most days, we can get samples with little additional cost to the fishery, and they can send them to us. And it's obviously someone's job to do every day, so it's not trivial, but it's a lot easier to get samples year round. And so using the fishing vessel, we're able to get a really high resolution of samples, which is quite unprecedented before. So I just wanted to give a little summary of the krill fishery, because I think it's a bit misinterpreted or misunderstood. So the fishery is managed by CCAMLR, the Convention for the Conservation of Antarctic Marine Living Resources. It's a bit of a mouthful, you can see why it's an acronym. And they're actually based in Hobart as well. And they use a precautionary ecosystem management approach to managing krill. And most of their decisions are also carried out by consensus. So everybody at the meetings has to agree for there to be a change to a fishery. So the CCAMLR oversees the krill fishery, but also other fisheries such as the toothfish fishery as well. And there's approximately 60.3 million tonnes of krill in Area 48. So this is where the fishing is confined to this area here. And that's because most of the krill are found in this area. And they currently have a total allowable catch of 5.6 million tonnes for the whole Southwest Atlantic Ocean here. And that's currently 10% of the krill in this area. But the real catch limit, I guess you could say, is the trigger level, which is 620,000 metric tonnes. So that's 1%. So that would apply to these two krill here. And if the catches get to this level, they need to be reviewed, and the CCAMLR needs to make sure that not all the catch is being confined to a small area. But at the present time, the krill fishery is actually catching far less than the trigger level. So they had the highest catch level last year, actually, that they've had in 29 years, and that was 390,000 tonnes. And this 390,000 tonnes also already gets split up between different areas within this fishing area to make sure that not all the krill has been collected in one spot. Because the main concern with any fishery is just to make sure that all the krill aren't getting caught in one area, in a concentrated area where there might be lots of predators that really rely on the krill. So there's some effort made to make sure that the catch is spread out as well. So the fishery is currently managed really well. In terms of many fisheries in the world, it's actually quite gold standard. And I think just spreading out the catch will remain the key focus into the future. And so you're probably wondering what the krill fisheries are catching krill for. It's mostly either for fish meal or for nutraceutical products, so those krill oil products that you might have seen in the pharmacies. So I'm going to start, just give you a quick summary on the methods that we used to look at the krill diet. So we had about 391 krill samples that I looked at. So the krill fishery collected 20 per day for us, but we just used a smaller sample because it would just be crazy to analyze every single krill. It would probably be 5 PhDs to do that. So to look at the amount of lipid in the krill, we extracted them using a range of solvents in the lab. And you can get, this is some vials here are some of the krill lipids. So it's got that nice red color due to the astaxanthin in the krill, and they get that from the algae that they consume as well. It's a pigment. Then we take these total lipid or total fats from each krill, and we go through another series in the lab. So there's a lot of glass vials and instruments involved. It looks very scientific when you're doing it, and it obviously is very scientific. And the samples get methylated, which liberates the fatty acids from the carbon backbone. And they go through a machine called a gas chromatograph, which separates the fatty acids. And then you get this nice little picture of the different fatty acids, and you get all the quantities and percentages of the fatty acids in the krill. So we can do that for each individual krill sample that we look at. And this map on the right here, where our samples were taken for my work. So you can see that there's samples from the 4 different seasons, and they are spread out between different locations. The fishing boat moves through, starts off at the Antarctic Peninsula in autumn and summer and moves towards South Georgia in the winter and spring. And over here on the left is just showing you the different areas where the fishing vessel goes, and you can see the whole continent there as well, just to give you an idea of the location. So onto some results. I know everyone loves graphs, so here some for you. So you can see that the total amount of fat in the krill, we saw this amazing pattern where it increased through summer into winter and decreased through winter into spring. And these are the 3 different years where the krill were collected for me. So we basically had 3 years of continuous data across all the seasons, which was really amazing, as we hadn't been able to see it at the scale before. And you can see the pattern's very consistent among the years studied. So you got the increase in fat stores in autumn and a decrease in winter and spring, and that happens across each of the years pretty consistently. And that relates to what the krill are feeding on. So as the phytoplankton blooms in summer, they increase their amount of feeding, and they get to that peak in autumn. Then in winter, when you get to the 24-hour darkness, the amount of phytoplankton decreases, and you can see their fat stores decrease as well, down to an early spring low. And some of the fatty acids show a similar pattern. So when I extracted the lipids and fatty acids from the krill in that process I described earlier, we actually found 59 fatty acids in the krill, which is obviously a lot. So I won't talk about all of those today. I'll just touch on a few, including the important essential omega-3 fatty acids that had the highest quantities in the krill. So you can see that same kind of seasonal trend, but in 2016 autumn, you see really high levels of this omega-3 fatty acid, EPA, in the krill. And that's, as I mentioned earlier, a diatom algae biomarker. So we can kind of tell from this that maybe in 2016, the krill were feeding on a lot more diatoms in the autumn season. And another important omega-3 fatty acid in high quantities in krill is DHA, and this is a flagellate algae biomarker. So we can see the same kind of seasonal fluctuations in DHA as well. And based on this information, you would also can see that the fishery would probably like to fish mostly in autumn when the levels of DHA are highest. And also potentially that they're most nutritious for predators at this time as well. This was a really interesting fatty acid. This is also a flagellate algae biomarker. And you can see that the seasonal trend is less pronounced. So it really increases in autumn in 2015 and also in 2016. But in 2014, you can see it's not, you don't get that real peak. It's more flattened out. So this one's quite important because the other two fatty acids I mentioned, they are conserved quite strongly by the krill because they're really important components of cell membranes. But something like this fatty acid SDA is readily replaced when the diet changes. So it's arguably a better indicator of diet than EPA and DHA. But one of the really interesting things was that the krill fishery, their specifications for SDA and their krill oil products, they actually managed to get those altered through some of my results from some of my research because we were able to show that it is so variable, and some of their specifications might have been too narrow for that. So that was quite a cool outcome of my research. All right, so I'll just sum up. That was the first couple of chapters of my thesis. So I'll just sum up the results from that. So we found that krill were predominantly herbivorous in summer, mainly feeding on diatom microalgae. Their level of omnivory increases as the seasons progress from autumn to spring. And so that's due to basically there's less algae when there's less light in winter and also in early spring. So they start to feed on other prey such as copepods during that time. But they do feed on copepods year round. It just increases during these seasons. They nibble on bacteria all year round. And we found some sea ice algae present in the diet of krill in some locations. So that was using another thing called some sterols that tell us that they're feeding on these diatom sea ice algae, which was interesting. And as you can see, the seasonal trends were strong, but there were some interannual and spatial differences that we could see as well. And it's really neat because although my PhD is finished, my supervisor Patty and some of her team now are being able to get some more data and follow the research on. So we've got, they must have 5 or 6 years worth of fatty acid data now. So it's really exciting to carry on that work. All right, so we know that krill are well adapted to life in their extreme environment at seasonal and interannual scales, as you saw. But we really want to know how resilient they are in an environment that is also now changing rapidly. As we know, humans are altering the Earth's climate. I probably don't need to tell all of you who are tuning into a krill talk, you'll know a lot about climate change already, but I don't think it hurts for us to see these graphs either. And this basically shows we've got carbon dioxide over the last 800,000 years, and it's increased higher than any levels over that time currently, and you can see the steep increase in the last, since the Industrial Revolution. And we've got this graph on the right here. As you can see, atmospheric CO2 is increasing, and it's increasing in line with our carbon dioxide emissions as well. So just on a side note, I'm reading this book at the moment called Hope in Hell, A Decade to Confront the Climate Emergency. It sounds pretty depressing, but it is what it says. It gives you quite a lot of hope for solutions for now and into the future. And I would recommend that to anyone who wants to read something that's kind of reasonably accessible for the public. And of course, if you haven't seen this David Attenborough documentary, who doesn't love David Attenborough? It's a really good one as well, talking about climate change and what we might expect to see in the future. This paper is one of my favorite papers of all time. It's kind of a choose your own adventure type story. I think it's open access, so anyone can download it, but I'd highly recommend it if you want to know more about what might happen in Antarctica with climate change. but they essentially present two narratives for the future of Antarctica in the Southern Ocean. One future where we do something about our carbon emissions and there's effective policy and governance. And another scenario with high emissions if nothing is basically done to curb our emissions. So you can see there's a lot of differences between these two scenarios. The air temperature increases, the sea level increases. I've got a range of things here. I won't go through all of them, but you can highly recommend reading this paper if you have the chance. So my PhD aim was to understand how adult Antarctic krill may respond to ocean acidification using the world's first long-term lab experiment on this topic. So what is ocean acidification exactly? I'm sure many of you will have heard of this. Back when I did my master's research, like 12 years ago now, it wasn't really common knowledge in the general public, but I think almost everybody has heard of it now. But as we all know, anthropogenic carbon dioxide emissions are increasing, and approximately 30% of those emissions are drawn down into ocean surface waters, where they bind with H2O to form carbonic acid. And this quickly dissolves into hydrogen ions and bicarbonate ions, and this increase in hydrogen ions lowers the pH of seawater. And this makes it difficult for calcifying organisms to use carbonate to build their shells and skeletons, because the hydrogen ions also bind with carbonate to form more bicarbonate. And another consequence of this process is that carbon dioxide also diffuses across into the bodies of organisms. So it diffuses across their cell membranes and into their intracellular and extracellular spaces, causing basically ocean acidification inside the body fluids. And currently we've got seawater carbon dioxide at approximately 400 parts per million in 2019, which corresponds to a seawater pH of 8. And it's predicted to increase to 1,500 parts per million and 2,000 parts per million by the years 2100 and 2300. And you can see that decreasing in seawater pH that corresponds to this happening. And acidification is unfortunately occurring fastest in areas with colder waters, such as the Southern Ocean, as carbon dioxide dissolves more easily into this colder water. So what might this do to Antarctic krill? And what do we know already? We know that egg hatch rates decline at carbon dioxide levels predicted for 2100, and that no hatching occurs at levels predicted for 2300. And for adult krill, we don't really know too much yet. So before my PhD research, there was one 24-hour study, and that found that krill increased their feeding and their nutrient excretion rates at carbon dioxide levels predicted for around kind of 2070, 2080. And there was a behavioural experiment done actually at the same time that I was doing my PhD research, and it found that krill did not discriminate between ambient and high CO2 seawater in a behavioural experiment. So whether they could discriminate between the CO2 levels and they didn't really mind too much, or whether they couldn't discriminate between the levels, we don't know. But a really interesting study that was done at the Australian Antarctic Division Krill Lab as well. We know from previous studies that crustaceans are generally more resilient to acidification than other invertebrates. So you can see here that the negative effects, most of the negative effects only occur at these really, really high carbon dioxide levels that aren't generally, they won't be seen in the real world. And we don't know exactly why crustaceans show this resilience, but we think it's most likely due to their generally active lifestyles and an increased capacity to control acidosis in their bodies. And that's through the transport of ions across their gill epithelia to control their acid-base balance. So many studies on other species of crustaceans have found that they have a higher ability to control this acid-base balance compared to more sedentary things like mollusks and echinoderms. So I've kind of called this the acid-base balancing act. So as carbon dioxide diffuses across the cell membranes of organisms and into their body cavities or extracellular spaces. They can correspond to this by pumping bicarbonate ions into their fluids. And that increases or kind of offsets the decrease in pH that occurs when the carbon dioxide diffuses across their cell membranes. So they can actively pump these ions across into their bodies, and the degree to which they are able to do this varies between species. And as I said, the active lifestyle of crustaceans indicates that they're better at moving these ions inside and out of their bodies. And it's important to keep the pH of their body fluids, such as their blood, at a certain level, because it's important for a range of different protein functions that control things like reproduction and growth. So we'll come back to this process a little bit later on. So in order to look at the effects of ocean acidification on krill, as I said, I ran a long-term experiment. So that went from January to December 2016. And I reared krill in 5 different carbon dioxide levels. So we had a present day, or as we call in science, a control, so a level that we currently see for a comparison. So I guess normal seawater, you could say. Then we had 2 levels of near future ocean acidification. So we basically bubbled carbon dioxide into the seawater like you in the same way that you would see in the ocean. And we looked at 3 different carbon dioxide levels for near future, and then an extreme level, which is not relevant in the real world. And that was at 4,000 parts per million, but we just wanted to see if the krill were really resilient. We just wanted to push them a little bit to see at what kind of levels they might show some negative effects. So that corresponds to a seawater pH of 7.1, which, as I said, is not environmentally realistic. If the oceans got to this level, we'd be pretty worried. So these are just some pictures of the krill lab. It's quite hard to take photos in there because it's not very big. But these are some of the tanks we used, and you might be able to make out some krill in the tank there as well. And we used a photo period, so we mimicked the light regime in the lab like you would see in the wild, to keep that as close as possible to the wild. And I basically did a whole lot of daily monitoring, looking at seawater pH, carbon dioxide, checking for any mortalities and molts. So kind of animal husbandry to make sure that I was collecting samples and the krill were as happy as they can be. And we fed them every day as well. So down at the Antarctic division, they actually grow Antarctic microalgae, so we can keep their diet as close as we kind of can in a lab situation to what they would normally eat in the wild. And I measured a range of different things across the year. So we'll go through some of these in the next slides, but some of these were measured regularly across the year, and some of them were measured just one time during the experiment. All right, and we found some really fascinating things in this experiment. So the first interesting thing was that despite being fed a constant and consistent diet across the year of the microalgae, krill actually stored fat in a similar way to what they would in the wild. So triacylglycerol here, don't worry too much about that. It's a pretty big word, but it basically means the fat stores like the total lipid in the krill. So you can see here in the summer months, we've got the different colors of the different carbon dioxide levels in the seawater. Their fat levels are lower, but once they get into winter and spring, they actually increase their fat stores just like they do in the wild through autumn into winter and spring. Which was interesting because we fed them the same amount of food throughout the experiment. So this kind of showed that what we think might be happening is that light cycle, or the biological clock that I mentioned, they're increasing that fat, expecting over for the winter, basically storing their fat for the winter like they would in the wild. But you can see down in this yellow one here, which is the highest, most extreme CO2 level, they weren't able to store that fat as efficiently as the krill and the other carbon dioxide treatments. Oop, wrong way. We also found that the length of krill over the year, it actually decreased. The median length of krill in all treatments actually decreased in winter and between summer and winter, which was really interesting because that's also what you would see in the wild. So you're kind of seeing that possible shrinkage happening. And that could be related to the biological clock again, when they decrease their metabolic rate in winter, and it causes that decrease in size. And you can see in the most extreme carbon dioxide treatment again, that they're quite a lot smaller. So not really seeing any major effects of the near future carbon dioxide, but we are seeing some effects of that extreme level of CO2. The respiration rate, so this is another word for oxygen consumption of the krill. We were only able to measure this once during the trial, so this was done in October, kind of near the end of the trial. So we did that by placing individual krill in these little tanks and used fiber optic sensors to measure the change in oxygen concentrations over a 24-hour period. We didn't really see any major differences in the respiration rate at these different carbon dioxide levels, but we did only measure this once in October. And they had already been in the carbon dioxide treatments for kind of 10 months by that point. So it's possible they may have acclimated during that time. But in future studies, it would be really interesting to look at the metabolic rate more regularly throughout the year as well. In terms of their reproduction status or their sexual maturation, we didn't really see a high effect of carbon dioxide. We didn't see any effect of near future levels of CO2 on their maturity, which we did this by looking at their external sex organs and looking whether they had developed in line how you would expect at the time of year in spring when we looked at the krill. And we looked at some ovary stages of the females as well to see if they'd progressed in the way that they should be progressing, and we saw at near future carbon dioxide levels for the ovaries and for the maturity score that they were progressing as expected. But again, in the extreme carbon dioxide level, their maturity level and ovary development was really delayed. So we're seeing an effect there. I won't go into this part in detail because this actually was a whole chapter of my PhD, but it's just highlighting the fact that fatty acids, they can tell us a lot about the diet, but they can also tell us quite a lot about the immune system of the krill as well. So we can look at the change in ratios of omega-6 to omega-3 fatty acids, and if you're seeing an increase in the omega-6 fatty acids, that's an important long-chain polyunsaturated fatty acid that is the primary precursor of things called eicosanoids, and they're molecules that regulate the immune system, the reproductive processes, and interesting, the ion flux of the body as well. So in the highest extreme CO2 level, you saw this change where some inflammation is possibly occurring in the krill. Again, we didn't really see any difference between those near future carbon dioxide levels though. One of the most important factors obviously is survival. Again, we didn't really see a large effect of carbon dioxide on survival. What we did actually interestingly see was that survival increases slightly in those near future carbon dioxide levels compared with ambient seawater. This bottom one here is that extreme level, so we saw a pretty large quick decrease in survival there. So we're not sure why we saw a slightly elevated level of survival in those krill in the near future CO2 levels, but this isn't totally unusual. It has been seen in other krill species before, but we think it might be one possible explanation is that krill exposed to vertically changing carbon dioxide in the water column may use that acid-base regulation that I mentioned earlier and short-term metabolic depression to enhance their survival under these higher CO2 conditions. Another thing can be that potentially the food levels in the tanks, because they are phytoplankton and they photosynthesize, using carbon dioxide, potentially their food levels have increased slightly in those tanks as well, but further research is really needed to understand the mechanisms behind that increased survival. So finally, you've got the message that adult krill hopefully are quite resilient to near future carbon dioxide levels based on those parameters that I've mentioned. And I think this kind of sums up why. So I'm hypothesizing that this might be due to their ability to control the acid-base balance. So here we have results of the hemolymph pH, or the blood pH of krill. And you can see on the left axis here, this is the blood pH. And on the x-axis, we have got the corresponding seawater pH in those different carbon dioxide treatments. So krill in ambient seawater, they have, their blood pH is quite similar to the pH of the seawater that they're in. Krill in the higher 1000 parts per million CO2 treatment, they've got a blood pH similar to krill in that ambient seawater, but to maintain that, they've had to increase their blood pH 0.3 units above that of their seawater pH. And in these slightly higher, again, CO2 levels, they've kept their blood pH again within that same range as those ambient krill. But to do that, they've had to increase their blood pH by 0.3 and 0.5 pH units. So this all requires energy to keep this blood pH at the correct level. And you can see in that 4000 parts per million treatment, that extreme level that I mentioned is not realistic in the real world. They haven't been able to maintain their blood pH the same as the other krill, and it has required quite a bit of energy to do that. Okay, so in summary, Antarctic krill were resilient to simulated near future acidification, but we don't know what the longer term costs are. So we don't know how the krill will have fared past that 1 year that I ran the experiment for. The krill in my experiment were quite well fed. So in the wild, where you have seasonally variable carbon dioxide concentrations, and they may not be able, if the food levels help them to survive better in higher CO2 seawater, they may not fare so well in the wild if they have lower levels. That metabolic depression that I was talking about, that's controlled by that biological clock, and increasing severity of winter acidification. So in the Southern Ocean in winter, there's a higher frequency of storms and mixing of the seawater, which brings up carbon dioxide, that's seawater that's higher in carbon dioxide to the surface, and creates a more intense acidification in winter. And the regionally variable food concentrations I just mentioned, they may increase the vulnerability of krill. So I think the winter months are a bit of a bottleneck for krill. And those endogenous rhythms that I mentioned, where they decrease their metabolism due to the light cycle, obviously the light cycle won't really change so much with climate change. So although they're very flexible, they've got these endogenous rhythms that are kind of a little bit inflexible. So it'll be interesting to see how those things mix together as the climate changes. And of course, we only looked at acidification in my experiment, so we don't yet know how acidification may interact with other stresses such as warming and changing ice conditions. So just to finish up, I mentioned earlier that we know that krill have vulnerable at the earlier life stages, so the egg and embryo stages, to acidification. But in my study, we kind of saw that the adults are quite resilient. So what happens in the middle here if these eggs can't grow into the resilient adults? So if I had free reign on the krill lab, if I was able to go in there now and do whatever experiment I wanted, this is the one I would do. So I would have been really interesting to take those resilient adults from my trial and keep them through the spawning period into that following December, January, and to see whether they did produce resilient eggs. And we've seen that in other species, such as in this study here, in Antarctic sea urchin, that we see those transgenerational effects, or the resilient adults producing resilient offspring. So if this is the case, then the embryos and eggs may not be as vulnerable as we think, but we just don't know if that's the case yet. So that's kind of the next stage of research to look at. So my final thoughts, my thesis results highlight the remarkable adaptability of krill in a changing environment, from short-term seasonal or annual scales to longer-term decadal scales. Their flexible lifestyle may aid their survival in an ocean that's rapidly changing with increasing anthropogenic carbon dioxide emissions. Future research investigating effects of multiple stresses on krill and transgenerational responses to these stresses will further our understanding of krill resilience to climate change. And I've just put this little figure here just to emphasize the fact that there's so many other things going on in their environment as well, multiple stresses happening that they're also facing. So it could be a challenging time in the future. And just to sum up, here's some further resources and references. So I swear my supervisor, Steve, didn't pay me to say this, but he wrote a really interesting book, The Curious Life of Krill. It's accessible to the general public, and it's I found it really fascinating. I'm biased, obviously, but really recommend that book, and of course the David Attenborough Netflix documentary. And if you're super keen, you can have a look at any of my papers and some of it with the research that I've talked about today. Some of them are open access, so anyone can access them, but some of them you might have to ask me for, but you're welcome to email me on this email address below if you want any more information. Thank you very much. I'll pass it over to Mary again.
Thank you very much, Jessica, for that very impressive talk. And I now invite all the people joining us today to ask Jessica a question by typing it into the Q&A button at the bottom of your screen. Now, first question for you today, Jessica. Do you think ocean acidification will affect the krill carapace in the same way that it affects shellfish skeletons?
Good question, Mary. That's something we haven't looked at yet. The krill carapace is mainly made up of chitin, so it's not. It's got a little bit of calcium carbonate in it, so it may indeed affect some of the skeleton, but it's not expected to be as much of a concern as things such as shellfish like mussels and oysters. So yeah, something interesting to look into, but I wouldn't imagine it would be a huge problem for krill.
Thank you, Jessica. Next question. Can you be sure that the patterns in fatty acids in the krill sampled by the fishery relate to the seasons and not the different sampling locations?
Sure, that's a really good point. So we did look at some locational differences as well. So we broke down some of the fatty acid data into locations and also into seasonal data. And there were some locational differences that we did see. But the main driver behind it seems to be the seasons. And we kind of know that too because it matches up really well with data from other studies that have caught krill in the same season, but from a different location. So we think it's mainly a seasonal driver, but certainly there are some finer scale spatial differences as well. And also, some of the locations are quite different. So South Georgia, for example, doesn't have sea ice around it, but obviously on the West Antarctic Peninsula, you get sea ice changing quite seasonally. So you'll get things like sea ice algae coming in a bit more in that area.
Thank you, Jessica. And next question, you mentioned the large biomass of krill. Given the large biomass of krill, do they produce waste that affects the environment the same way that cattle do?
Oh, that's a very good question. Well, a lot of any waste that krill excrete will kind of sinks down to the ocean floor, I guess. Whether it makes it all the way or disintegrates kind of on its way down is kind of not my area of expertise. But no, I wouldn't think it would. It fertilizes the ocean surely, but it's not. It doesn't pollute the oceans. If that's what you're suggesting, I guess.
Thank you, Jessica. And I'm sure that many people listening today have questions that they would like to ask Jessica we haven't got time for today, or that they think of later. And Jessica's put her Twitter handle on the screen so you can follow up with Jessica after the lecture. And I'd now like to introduce our Royal Society of Tasmania Vice President, Professor Jocelyn McPhee, who chairs our Honours Committee. And Jocelyn will propose a vote of thanks to Jessica. Thank you, Jocelyn.
Hello, everyone. Jess, I'm sure everybody in the audience will join me in thanking you for all the time and effort you've put into your lecture today. I know you've got a lot on right now, and it's really appreciated that you made yourself available. I was pleased to hear you didn't mind the online format, because it certainly does present some extra challenges, but you handled it superbly. So finally, our thanks this afternoon, and congratulations on winning the Doctoral Awards. I now totally understand why. So would you like to make any final comment before we hand back to Mary?
Yeah, I'd just like to say thank you again to everyone who tuned in. There's a lot of other things going on in the world right now that could have occupied your attention rather than my talk, so I really appreciate it. I hope you all learned something new about krill. And yeah, I really enjoyed the webinar. It's quite unusual talking to an empty room, not being able to have any audience reaction. Actually, it was a bit more difficult than I thought, but I really enjoyed the talk, and thank you again for letting me share my research today.
Well, Jessica, on behalf of the Royal Society of Tasmania, I thank you again very much for that really impressive talk. And I'm sure it gives the people listening today an indication of the calibre of nominations that we get for our doctoral awards. And just a quick reminder that it's not too late to nominate somebody for the 2020 doctoral awards. They close on the 15th of November, and all of the information is on our website, which is on the screen in front of you. And I'd like to invite you to our next lecture, which is going to be in person. It's going to be Antarctic historian John Williamson talking about the development of Hobart as an Antarctic gateway and what the historical record tells us, the role of Roald Amundsen's expedition. That'll be fascinating. So that's on the 30th November at the Royal Yacht Club of Tasmania, and that's our Christmas dinner lecture. So all of the information about that is also on our website, rst.org.au. Now, sometimes people wonder if they're welcome to come along to our events. So our events are not just for members, they are also for our friends and supporters. So you are all very warmly invited to join us for that event. If you have any questions about the Royal Society or any feedback for us, please email us on the email address on the screen in front of you at royal.society@tmag.tas.gov.au. And if you're not already a member of the Royal Society, we ask you to consider joining, because membership is open to all people with an interest in the advancement of knowledge. And I'm pretty sure that that's everybody listening in today. We've been extremely pleased this year that even during the pandemic, every single month we've had new people join us. So we are always delighted to welcome new members. And in closing, I'd like to thank all of the people who helped to prepare and promote the webinar, and as well as our fantastic speaker, Jessica. So thank you all very much for joining us. Please keep an eye on our website. We've got our 2021 program in mind, and there's some very exciting events coming up, starting with our annual general meeting on the 7th of March next year, at which the speaker will be Tasmanian Australian of the Year, Dr. Jess Melbourne Thomas. So there'll be many other exciting events for 2021, and we hope you can join us for some of those. Thank you for joining us today.